Alright, welcome back. In this video, we're going to go over steps 7 and 8 in gluconeogenesis. So at the end of step 6, we got glyceraldehyde 3-phosphate. Now we're going to take that and do a little intermediate reaction. So we technically actually have 2 molecules of glyceraldehyde 3-phosphate. Because in glycolysis, at the end, we created 2 molecules of pyruvate. So that means we technically actually have 2 molecules of everything going on since then, since the beginning of gluconeogenesis, because we're going backwards. So we actually have 2 molecules of glyceraldehyde 3-phosphate. And here's what we're going to do. We're going to take one of those molecules, and we're going to use the enzyme triose phosphate isomerase. And what this enzyme is going to do is going to convert the aldehyde, one of those aldehydes that we have, the glyceraldehyde 3-phosphate, into an alcohol. And this alcohol will be called dihydroxyacetone phosphate. So now at the end of this, we're going to have 1 molecule of dihydroxyacetone phosphate, which is the alcohol. And then the other molecule will stay the same as glyceraldehyde 3-phosphate, which is the aldehyde. So here's what this enzyme is going to do. All we're going to do is we're going to switch the places of the double bonded oxygen and the hydroxyl group. So in the aldehyde, the double bonded oxygen was on carbon number 1. The carbon number 2 had the hydroxy group. Here's what we're going to do. We're going to swap it. So now the hydroxyl group is in carbon number 1, and then the double bonded oxygen is in carbon number 2. That's it. Pretty simple. Now step 8. Step 8, we have 1 molecule of the glyceraldehyde 3-phosphate, which is the aldehyde, and 1 molecule of the alcohol, the dihydroxyacetone phosphate. And we're going to use an aldose enzyme. The enzyme is called aldose. And what we're going to do is we're going to fuse these molecules together. Now the phosphates on both molecules are going to be the tails. So what we're going to do is we're going to put the alcohol, the dihydroxyacetone phosphate, on the top, and then the aldehyde is going to sit underneath it and it's going to join. It's going to join between carbon number 3 and carbon number 4. So the phosphate groups sit on top and on bottom. So those are going to be carbon number 1. And then the carbon number 6 will be the other phosphate. Then all we have to do is the carbon that is 4. So what I did is I actually I numbered these carbons and how we're going to stack them. So notice that the glyceraldehyde 3-phosphate here is labeled carbon number 4, carbon number 5, and carbon number 6. Even though it's technically not true, I did it for simplicity. So you know how it lines up in the full molecule. So the only thing we're going to do here is first stack it up. So the alcohol is on the top and the aldehyde is on the bottom, with the phosphates pointing down and up. Then the double bonded oxygen on carbon number 4 is going to change into a hydroxyl group. And that's it. It's that simple. It looks complicated. It looks kind of messy and complicated, but it really isn't. All you're just doing is putting the alcohol on the top and bond it together with the aldehyde on the bottom. The phosphate from the alcohol points up, the phosphate from the aldehyde points down, and we change the double bonded oxygen on carbon number 4 to a hydroxyl group. And that's it. So if you find this video helpful, please like and subscribe. Until next time, later.